Welcome, everyone. We're excited to have you here for our webinar on navigating complexity in PROTACs with PBPK modeling. And we hope that you gain valuable insights and practical takeaways from today's session. Feel free to engage throughout the presentation, but also note that we will address all questions at the end of the session. So before we get started, I'd like to begin with a brief introduction to Solvita and to today's speakers. Within drug discovery, we provide at Solvita integrated solutions supporting clients in advancing their research from target identification to preclinical development. We leverage expertise across multiple scientific disciplines to progress projects and pioneer innovations in key disease areas, which include oncology, inflammation, fibrosis, respiratory, metabolic, and infectious diseases. And having said that, I would like to introduce our speakers today, who are my colleagues from the Integrated Drug Discovery team, David and Simone, both very passionate about applying PBPK modeling concepts in early discovery. David Cebrian brings over 17 years of experience in both academia and industry. His expertise spans PBPK and PKPD modeling with a strong focus on translational PK in oncology and infectious diseases. He has contributed to 5 clinical candidate nominations and co-authored more than 15 scientific publications. Joining David today is Simone Esposito with over 15 years of experience in drug metabolism, PK, and bioanalysis, particularly a lot of focus on peptides. His work combines experimental and modeling approaches, specializing in PBPK modeling for complex modalities such as PROTACs. And Simone has contributed to 4 clinical candidates and authored over 25 publications. With that, let's dive into today's webinar, and we'll follow on with the Q&A at the end of the presentation. Simone.
Thank you very much, Jasna, and welcome everyone. Today we are going to share with you our experience with applying PBPK modeling in for the understanding and the prediction of pharmacokinetics of products. We'll be starting with an introduction over the ADME challenges that we encountered in product projects and the need for an appropriate ADME integration. And then we will be introducing the PBPK modeling, in particular in the context of oral absorption for beyond a rule of 5 molecule. Then we will go through 2 case studies. The first one that will use the Arvinas 110 as a tool compound to show how a PBPK model for product can be built to understand its pharmacokinetics. But a second case studies will be focused on the impact of pH, gastrointestinal pH, on product oral absorption, and we will be using the Nurix 127 compound as a tool compound. So products require really little introduction. They are probably the most popular, surely the most advanced class of bifunctional molecules inducing selective protein degradation, and they link protein target with the E3 ubiquitin ligase component. They promise to be a good alternative for particularly difficult to target targets, and they all promise also to achieve low dose, thanks to high potency and selectivity, but also to the particular mode of action that is more event-driven rather than occupancy-driven. The first product to hit the clinic was the Arvinas 110 in 2019. There are currently more than 20 products in being tested in clinical trials, with the most advanced being the Arvinas 471, which entered phase 3 clinical trials 2022, and just a few weeks ago, some first positive data were released. So the products hold a lot of, are very attractive from the point of view of selectivity and potency, but from the DMPK perspective, they present some very complex challenges. Being beyond rule of 5 due to the particular structure based on 3 components, they tend to have high molecular weight, a high count of hydrogen bond acceptor and donors, as well as a high lipophilicity. And these extreme features are impact, of course, also their DMPK properties from physiochem and ADME, pharmacokinetic, and leading, for example, to a molecule with poor solubility, low permeability, that translate often to low bioavailability, but also difficult in vitro in vivo or challenging in vitro in vivo correlation, in vitro extrapolation that make it the prediction of human PK highly uncertain. And not only the property themselves are of complex interpretation, but also their measurement present complex challenges. Everybody who works with PROTACs is familiar with when running DMPK assay. Very often we observe low assay recovery or limitation of sensitivity. In some assays, low incubation times are required, and in general, the whole DMPK screening cascade is affected by a low throughput. So, and basically, here we get to the statement of our problem. So we actually acquire a lot of in silico, in vitro, in vivo data for PROTACs, but their interpretation is very difficult, making their optimization difficult or at least less effective. Here we show some of the questions that we typically, many of you, I'm sure, are familiar with that occur into the PROTACs project. For example, how this compound can be orally available if they have such a bad in vitro ADME profile, or what permeability assay should we rely on? Should we use MEMDCK? Should we use CACO2? Should we modify this assay? Also, some properties are intrinsically difficult to measure. PROTACs typically have very high PBB, and sometimes the typical assay, but also other alternative assay faces great challenges in measuring them. Typically, chemists use multi-parameter optimization methods, and recently some literature has shown that some of these MPO algorithms were also developed for PROTACs, but it's very difficult to apply to different chemical series. And of course, from a PK and PKPD perspective, we would like to know which is the species that is closer to human to predict human pharmacokinetic and human dose. So when facing all these difficulties, we definitely need a framework that allows an appropriate data integration, an appropriate DMPK data integration. And we think that PBPK modeling is a very valuable tool in this context. So, for example, speaking of the absorption challenge with PROTACs, we know that with PROTACs, while achieving a low hepatic clearance, so a high fraction passing the liver is achievable. But in many cases, the fraction absorbed is the one that is limiting the absorption. Of course, absorption depends on a number of physicochemical and ADME properties that we try to predict these properties with the in vitro and in silico, with in vitro assays and in vitro and in silico predictions. But we are well aware also that this process is highly dependent also on the species-specific anatomy and physiology. For this reason, PBPK, physiological-based pharmacokinetic modeling, seems to us to provide an ideal mathematical framework to integrate both physiochem and ADME data and the physiological parameters that are relevant for absorption. In general, we believe that implementing PBPK approaches is a valid approach for all the modalities, but we think that for PROTAC, this would really have added value. And now David will provide us with a brief overview on what PBPK brings to the table.
Thank you, Simone, and Yeah, Simone has nicely set up the scene, so it's going to be easier for me to define how this framework, this physiological-based pharmacokinetic models can help into the to dig into the main issues, especially regarding oral absorption as this complex physical space. So basically, and as you can see in this slide, if we focus on the BCS classification, these molecules would generally fall into the category number 4, which means that they come with low solubility and low permeability. But obviously, this doesn't mean that this space is not developable. As you can see in this table here, there are a number of compounds that belong to this class number 4 and still have been or have reached the clinic. So the thing comes to understand what are the complexities and the main issues to achieve relevant absorption for achieving the efficacy. So for this kind of molecules, obviously, the time frame for the absorption, along with anatomical and physiological conditions in the different segments of the gut, and obviously across various species, would be even more important than for classical rule of 5 space. So in this regard, this kind of framework, mathematical framework for physiological-based pharmacokinetic model incorporate specific information relevant to the absorption of these compounds. And we will be focusing here, as Simone has mentioned before, oral absorption. So that means removing the part of the impact of first pass metabolism and what happens after the absorption to the portal vein. So we will be focusing on what happens in the enterocyte barrier. So in this regard, factors like pH, the surface area to volume ratio, transit times, and length would be important in terms of providing the time frame that these molecules need to get absorbed due to their poor physicochemical properties, especially as we were mentioning at the beginning, solubility and the permeability. So bringing together both species-specific anatomical and physiological factors together with the compound-specific properties, this framework would allow us to interrogate compounds and understand which factors would be more sensitive and hence need to be optimized for these kinds of projects. So in the way these models are used, obviously there is an initial phase of validation, which we will go more in detail through the case studies. But after we get a validation of the model with existing data, then we can interrogate the different factors as I was mentioning. Obviously, there is an interplay between, as I said, the specific properties of the drug and physiological factors like the pH or the length of the different segments of the gut, the transit time, and area to surface, so area to volume ratio. And by incorporating this information into mechanistic equations, we can perform sensitivity analysis, which basically comes down to changes in the different parameters and understanding what those changes, how those changes impact, specific PK parameters, like in this case Cmax or UC after oral administration. And from that, we can come up with information of which parameters are key to focus on for the optimization of the PK behavior after oral administration of these molecules. So now we will try to give more detail with these 2 case studies that we are bringing on all these, the method validation of these models and also how we could apply the development of these kind of projects. So, Simone,
yeah, thanks again, David. So in this first case study, We would like to show how PBPK model for a PROTAC can be built to predict its pharmacokinetics, and then to translate this into an ad-based screening cascade and optimization workflow. Here we have used it as a tool compound, the Arvinas 110, but we can pretend to have a lead that will be it's a candidate for further optimization. And in this case, we will be, of course, exploring a number of assays, in vitro ADME assays, and in silico prediction tool to characterize this compound. But as you can see from the table on the right, despite having tried a number of assays, a number of protocol modification, in some properties are being really hard to measure or provide inconsistent data. For example, as you can see, MDCK in the standard and a standard setup, and CACO2 that we have tested that we used to for permeability, but in modifying, making multiple modification of the protocol, just provide values that were below the limit of quantitation. And in the case of lipophilicity, we have, in terms of value, the range is from 3.3 to 6.7. Some assays here failed, for example, the shake flask flow D and immobilized artificial membrane chromatography. So making difficult to understand which is the more reliable input for lipophilicity. Also, fraction unbound was higher and 99.9%. So again, below the limit of quantification of the assay, as well as compounds proved to be very stable, also both in microsomes and hepatocytes. So in order to apply this data in order to understand how this input and select which are the most informative assays or in silico prediction, we build a PBPK model to describe the pharmacokinetic in mouse and rat. For this compound, they showed a moderate bioavailability in both species. To do so, we have built first an IV PBPK model using in silico, in vitro, and in vivo data, but also combining, as we have already stressed, with the information related to species, physiological parameter, and we apply the different distribution models that are available in our PBPK software. And once we obtained, as you will see, a good fitting of the IV profile, we use the information from the IV to further develop a method to describe the PO data, which, by the way, are the type. Being the compound dosed orally will be the main question of interest to understand the PK, the PO of this compound. So, as already mentioned, the scope of developing the intravenous PBPK model was to validate the prediction of clearance and volume distribution that can be then applied to the PO model integrated with data that are relevant particularly to the absorption process. And here, as you can see, and I'll go into more detail in a few seconds, we managed to predict the profile of both species within twofold, with all the main IV parameter predicted within twofold. What did we do to tackle the absence or the total absence or the lack of robust measurement for some properties? So in absence of at this stage of in vitro clearance that could be then translated into vivo, we simply use the hepatic the total plasma clearance for this compound from the vivo PKs in rat and mouse. We have explored different KP model used for distribution, and finally found that the so-called PK-SIM standard model was the one providing best fit. And then we were left with a couple of properties which were really difficult to measure or were lacking robust measurements. So lipophilicity and PBPK and PBB. So for both properties, we have used a feature of PBPK modeling software, which is called parameter identification, that allows to estimate data that are uncertain or poorly measured, and we adjust these values to fit the IV profile. And as you can see from the table, we ended up with a value of lipophilicity value for lipophilicity was 4.7, and a value of fraction unbound plasma that was close to 99.9%. But as being this parameter particularly sensitive for determining the profile, the small variations in the fraction unbound, where the small adjustment in the fraction unbound were key to obtain a good fitting. Why, if we are showing also, for example, an example of wrong profile using chromulo D, which is sometimes told as a good prediction of a good method to assess lipophilicity, but in this case, using the experimental chromulo D values were causing a major over prediction of the volume of distribution. Once we got this good fitting for the IV data, we moved to the PO, and basically integrating the knowledge acquired from the IV model and incorporating incorporate the key parameter for assertion. In particular, here we have to say that the experimental values that as you already saw were either BLQ for MDCK and CACO2 or very low for Pampa, were largely underpredict the in vivo permeability, while permeability was better predicted with in silico predictions. And in this particular case, we also saw how the prediction of biorelevant media solubility, in particular in this case done with ADME predictor, was also catching well the profile for both mouse and rat. So, in conclusion, with this first case study, we have successfully integrated the data in silico, in vitro, and in vivo data for Arvinas 110, obtaining an accurate prediction of the PK in both rodents. We have tackled the absence of we have tackled the difficulties in measuring some properties by using either in silico predictions or parameter identification features. And this approach that was used with the two compound with available literature data can also, we think that it can be applied to virtually any PROTAC chemical series to understand their pharmacokinetics and predict particularly the PK of newly synthesized compound. So that's why typically when we start when a project starts to move forward, the product project starts to move forward, we always recommend to validate a PBPK model for a particular chemical series. And the way we typically do this is by selecting a number of representative compounds of this chemical series, generate in silico, in vitro, in vivo data the same way as it was shown for Arvinas 110. Use this data to develop and validate a PBPK model with, of course, a particular emphasis on the PO and on the absorption, but also taking into account also the other processes of the other ADME processes. And if successfully validated, then the PBPK model can be applied to new scenarios and to, in particular, to predict the PK of new compounds. So after these first case studies that was more focused on integration of drug-specific data, David will give us an example of how the importance of physiological parameter, particularly the pH of the gastrointestinal tract, are key to understand the absorption process of products.
So thank you again, Simone. So yeah, once Simone has nicely summarized the process of validation of these PBPK models, then I will focus on the interrogation of these models for the specific issue of oral absorption, in this case, PROTACs. So for this case study, we have used NORIX 2127. So basically, this is a novel bifunctional orally administered PROTAC or degrader, which induces the degradation of proton tyrosine kinase, and is currently in clinical phases. But basically, the question that we found interesting to show in this case study, and you can see in the table here, is the observation of decreasing bioavailabilities with higher species from rodents up to cyno, as you can see in the table here, falling from moderate bioavailability in mice to really low in cyno or dog. So we were interested in knowing if PBPK could shed some light on the reasons behind this observation, which obviously could be key in translation of PK to humans. And obviously, this is always a question mark, as Simone was mentioning in the introduction, about which species would be more relevant for prediction of PK in human for this kind of molecules. So in this case, we performed initially the validation with the intravenous available data, and what I'm showing here in this plot is the validation in of the oral administration of the NORIX compound. So as you can see in the plot, you see that both mice and cyno, the model is able to fit well to the observed data, as you can see when we compare visually the line and the dots, which are the experimental values. But also you can see in the table below how the values of Cmax and AUC nicely match for both species. Whereas in the case of the human, you see that there is initially kind of good fitting, initial part of the absorption, but then the model predicts a decrease, whereas the observed profile is flatter, as you can see by the dots. So from this observation, we interrogated the model to understand which could be the reason or reasons behind this behavior or the lack of prediction in human, whereas in the preclinical species using input data from lipophilicity, pKa, solubility, the permeability, and fraction amount in plasma, we got good predictions. So as you can see in these plots here, PBPK provides with functionalities to understand which are the concentrations in the different parts of the gut. In this case, as you can see here, we have summarized the concentrations in duodenum, jejunum, colon, obviously plasma, as you can see that we got this connection, and also excretion in fishes, which was significant. But mainly you can see that potentially this prediction of the model with a drop of concentration could be due to this dramatic drop in the concentration of duodenum and jejunum happening early after the administration. So then we perform a sensitivity analysis, so interrogate several aspects like the intestinal transit times, gastric emptying time, and also solubility predicted in the different parts of the gut to see which one came up as more sensitive, and it came out transit time in small intestine and gastric emptying time as the most relevant parameters. So we performed an optimization of these parameters, coming up with this plot in the right-hand side. And as you can see in this case, we've got a nice fitting of the plasma values to the observed values. And when you compare the duodenum and the jejunum concentrations, you see that they remain more time in solution as compared to the initial observation. So obviously, transit times is something that we cannot change. That is specific on different species. But what that means, so there could be potential explanations, like although it has not been disclosed, but potentially the formulation that could be or have been used in the clinic could be a controlled release, and so that would be explaining the slow absorption process. But what seems clear is that these kind of parameters, and especially solubility in the different parts of the gut, are governing the type of profile that we are observing in vivo. So if we look at the different parameters that the PBPK, this case PKC, which is the software that we have been using, provides, we see that there is a significant drop or shift in the pH, especially when moving from stomach to duodenum. And this shift, as you can see in this table here, is increasing with higher species. And obviously, it is increasing also the solubility ratio from the stomach to duodenum being greater with higher species. So this potential shift between the higher solubility in the stomach as compared to the duodenum, you see that there is a kind of a correlation with the observed oral bioavailability, which could explain somehow if a precipitation is occurring because of this dramatic shift in the solubility from stomach to duodenum, could explain this lack of or this decrease in absorption in higher species. So having that in mind, and with the conclusions of this solubility being in the different segments of the gut being key, and not only the solubility that we usually measure in the typical simulated intestinal and gastric fluids, but potentially a more dynamic assay providing information on the shift from pH and media composition from gastric simulated fluid to intestinal simulated fluid, could provide us with information on this aspect of potential precipitation when moving throughout the gut, and could be used for this kind of projects as more informative compared to the usual design of this kind of evaluation. And just as a summary, and to wrap up what we have discussed within this webinar, we wanted to convey the message of the importance of the use of PBPK model, especially with complex areas like the ones that poses these PROTAC projects. Validating these models early on the projects could provide with important information in terms of optimization. So basically, those are validated. So that means that some experimental data collection is required. Then we validate those models, interrogate those to come up with the identification of key parameters. And whenever we understand those, we can focus our experimental strategy on those that we consider are more important. And whenever we are producing more and more data, we can always improve the model. So it's like, let's call it a living creature. And we can end up with better validated PBPK models with outcomes like not only coming up with a more accurate oral absorption prediction, which helps on compound prioritization, but also to understand better which parameters need to be optimized and how. And also something that is key in the development of projects is to understand the risks and gaps that we face, and obviously put in place and design mitigation plans accordingly. So yeah, we hope that you found this content interesting. So now we will be happy to take any questions you may have. Thank you.
Thank you both, to David and to Simone, for this very insightful presentation and showing us examples of how you apply PBPK modeling in PROTAC discovery projects. As David said, we can now move into the Q&A session. If you haven't already, feel free to drop in some questions into the chat. Let me just check. So one of the first questions, if the PBPK model to predict the preclinical species PK profile requires the use of actual plasma clearance value following IV administration, how can you use it to predict exposure of compounds not yet administered in vivo to preclinical species?
Want me to take that?
Yeah. Sure.
So, clearly, it would be project dependent if clearance, can be predicted from in vitro systems, or in microsomes or hepatocytes. What we would recommend in that sense is that to have a possibility to run IV PK first to get the possibility to run IV PK first either as a single PK, discrete or cassette PK, for example, for an high throughput. That could be an idea to filter compound ahead of PO studies.
Another question, which other challenging modalities can benefit from PBPK modeling?
That's a good question. I think that these new modalities raising comes with challenges, same as PROTACs, with complex physical space and challenges in terms of distribution. Like, I don't know, I'm thinking about oligonucleotides or peptides with distribution issues or absorption challenges and so on. So I would say that any modality could benefit from this kind of PBPK models due to the fact that they incorporate all this physiological information into the equation, and obviously allows us to, or provides us with a framework to interrogate in a more informative manner the chemical series. And in fact, even for the classical small molecules within the rule of 5 has been proven as a great strategy to follow in terms of human PK prediction.
There's a question on, will we share the slides later? And yes, we will share the slides later. I have a question, David and Simona. So you talked a lot about the solubility and proposed some ways of measuring that. So are precipitation effects accounted for in absorption models? And can you use this modeling for optimizing formulation strategies to increase and enhance the solubility and absorption for PROTACs? Can you elaborate a bit on that?
Yeah, sure. Shall I take it? Yeah, it takes into account potential precipitation issues. Like, as I said, it takes into account pH in different segments of the gut. So that means that integrated that with the PK of sensitive drugs in this regard will predict a drop in solubility across the gut. So that could obviously come with precipitation issues. Also, the fact that it takes into account the solubility of the compound together with the solubility in the first segment of the gut where that the drug will be exposed to. So that also will take into account precipitation issues. And to the question of if that can be used to optimize or to understand the formulation strategies, then yes, there are different softwares that come with modules that allows to integrate even in vitro dissolution rates that could inform different formulations to come up with kind of rank order to those that are behaving better in terms of delivering the compound for absorption process. And then with in vivo data, you can always perform these in vitro correlation assessments to understand which, if you validate your in vitro model for the dissolution rate, and hence inform the strategy for formulation development and optimization. Yeah.
There's another question. How do we perform in vitro for this compound to use in IVC?
So, I mean, the short answer is that there's not the magic, trick. I mean, typically, we as a general comment, we tend to explore as many possibilities as possible. So different assays, different technique, different, and within this technique also different assay conditions. So the typical example of CACO2 with different pH, either the donor receptor or presence of BSA, or but our general message is that once we are focusing on a particular protochemical series, that we think it's mandatory to have this first exploration part and then link the most informative assays. I mean, understand which are the most informative assays to do IVC. I mean, generally, we think that it's not going to be an easy job in any cases. So this initial exploration is key to see what are the data that we can use. It's generally, I mean, true for all projects, but for this particular modality, we think that it's even more important.
Another question: when performing the sensitivity analysis, what would you class as significant?
Well, you've got a rank that the software will provide you with. So basically, what it does is iterating changes in the different parameters. So you just pick your let's call it in this case dependent variable, which would be the PK parameter, the Cmax or the AUC, and then iteratively the parameters that you have chosen to interrogate will iterate in a range that you define. And then obviously the software will compute the changes in Cmax and AUC, and obviously that will compute as more sensitive the ones that comes with a higher fold change in the PK parameters that you are interrogating. So that will be the way the software deals with this sensitivity analysis.
And just maybe for another aspect of, will you be working on other parts of applying PBPK modeling with PROTACs on anything else but what you've done so far, or what do you envision as the next steps?
Well, I mean, in terms of PBPK model exploration, will be good to acquire more and more data on multiple chemical series that will at least give us possibility to see as many different because having this complex approach, it will be good to reinforce it with multiple chemical series, I think. And then I think for now we have explored only the PK components of products in terms of, I mean, showing today. It would be also interesting to use PBPK model, integrated PBPK models with PD models to integrate both sides of pharmacokinetic and pharmacodynamics, because also in that. On the pharmacodynamic side, as also shown from by a few papers that have been published in the last couple of years, there is also a lot to learn, I would say.
Okay, I think we're heading toward the end of the webinar. I would like to thank you both for sharing this data with everyone. I would like to thank the audience for attendance. Feel free to approach us with any questions that might arise. The presentations will be sent through, and I wish you all a nice evening and a nice rest of the week.
Thank you.
Thank you, everyone.
Thank you.